Go ahead. Other questions?
I take back everything else I'm going to say after the time.
For Dr. Greek, my question is regarding polio. Could we have come about with or could a cure for polio have come about without animal experimentation? And if so, how long would it have taken?
Thank you. Well, again, according to Dr. Sabin, it would have taken 30 or 20 or 30 years less long than it did. Okay? Again, that's not Ray Greek, that's Al Sabin, who probably knows a thing or two about it. What animal experiments allowed us to do vis-a-vis polio was it allowed us to grow the virus, and it allowed us to test the purity of the vaccine. Okay? And again, if you go back to my second slide, where I talk about how animals are used in science and in research, animal tissue today is used ubiquitously to grow things. It's part of culture media. Now, we don't have to use animal tissue. We can use tissue from humans, of which there's a surplus. People die every day. Society finds that a little unsettling, and that's fine. But there's a difference between something being necessary and something being functional. And so the original polio vaccine was grown using kidney tissue from monkeys. Okay, fine, great. I mean, that's fine. What paved the way for the polio vaccine, you got to remember, Sabin and Salk didn't win the Nobel. Okay, the guys who won the Nobel were the guys who made the virus grow in vitro. That's what took the brains. Okay, I'm not saying Salk and Sabin were dummies, they're far smarter than me. But that's where the great breakthrough occurred. So it's disingenuous to say we studied polio in monkeys. No, we didn't. And when we did, we thought that polio entered through the nose and went to the CNS. That's what Sabin was talking about when he said it delayed the vaccine for decades. That was wrong. It went through the gut. Okay? So, we can't second guess history, but all of the ingredients were there many years before they were all used.
All right, please forget what you just heard. Sabin had been up to his dying day, had been very upset about how his testimony before Congress had been taken out of context and has been misused. He always defended animal research as having been essential for his work and for Sabin's work, Jonas Salk's work. The mistake that happened, by the way, it wasn't because of the animal studies that an error had been made. The error had been made not because they use animals, but because it was misunderstood. There was plenty of evidence already, going back quite a while, that showed that the gut did get infected. What happened was Sabin, in the late 30s, had conducted an experiment on fetal human tissue, not animals. He got a fetus, and he made cultures of the cells for different organs of the fetus. It was an aborted fetus. And unfortunately, he got a strain of the vaccine. Remember I said there's well over 100 strains of poliovirus that was expended throughout the world? He got one that was a laboratory specimen. It was grown and grown and grown, not in nature. It was grown in a laboratory for many, many generations. And it lost its ability to infect the gut. He didn't know that. So he used this strain from Rockefeller University and it did not infect anything but nerve tissue. It was an unlucky break. He could have used 100 different strains. He picked one that didn't infect nerve tissue. It was a mistake that set them back for about 14 years. Well, anyway, Dr. Greek blames that on animal studies. It had nothing to do with animal studies. In fact, it was grown in human tissues.
Perhaps I can also interject here. There was also some enmity between, or differences of opinion between Salk and Sabin. I knew Jonas Salk, and the difference of opinion was the use of inactivated virus versus a live attenuated virus. Sabin believed strongly in the latter, and Salk believed equally strongly in the former. Salk got there first, and that was the first vaccines that were used. And Sabin was in an unforgiving mood after that because he thought that Salk had got the priority for that. So there's an element of a scientific controversy. It's a fascinating history of two great men.
Can I ask for clarification? Because what I heard Dr. Grieg say is that Sabin said something about animal experimentation. What I hear Andrew Skolnick say is that Dr. Grieg made a statement about animal experimentation. And what you're saying by saying that is that Sabin didn't say that, and what you're saying is that Sabin said that. Yes, what you said was that what Dr. Grieg said this about animal experimentation, and Dr. Sabin believed and said something else, but what you said was that Sabin said it. So I just like a clarification of what did Sabin say.
His quote was accurate.
Yeah, Sabin said it. and that's all I'm saying. To get into how the polio vaccine came about, you know, we could go round and round on every, not just the polio vaccine, but every aspect of it. And I tell you, just to hopefully speed this debate along a little bit, look, for the sake of argument, I'm willing to give the animal experimentation industry every discovery up to the year 1990. Let's just assume for the rest of the day that every breakthrough in medicine up to the year 1990 was 100% dependent on animals. I don't care. That's not the point. Even if that were the case, the point is that in the year 2005, status post the Human Genome Project, status post the discovery of complexity theory, Evo, Devo, and so forth, folks, the name of the game has changed. Okay? So we can debate history till everybody's blue in the face, and it's not relevant to the argument that I am making.
I didn't say Sabin said that, but it was taken out of, it was, you know, he was speaking before Congress, it was taken out of context and misused. He had rebutted the misuse many, many times, and Dr. Grieg had been misrepresenting what was said, not misquoting. Anyway, the 1990, speaking of 1990, in Dr. Grieg's third book, he has a list of the last 20 years of Nobel Prizes. He leaves out 1990. When he was debating Dr. Speth on radio in 2002, Dr. Speth talked about kidney transplantation being essential using the dog model. Dr. Murray had won a Nobel Prize for that. Well, Dr. Grieg said, oh, Dr. Speth, you should know better than that, something to that effect. The first successful transplant was in humans. Was only successfully done in dogs 5 years after. Dr. Speth was dumbfounded. He didn't know enough to contradict him and say that's not true. Unfortunately, it was not true. Might be why Dr. Grieg did not put the year 2000 Nobel Prize in his book. Not 2000, 1992, because if you look at the 1992 Nobel Prize, it was to Murray for doing the first successful human transplant after developing the procedure in dogs. Check out, go to the website, if you doubt this, go to the website, look up the Nobel.org, and look up medicine, 1990. Dr. Murray had worked on dog model to perfect his technique.
Further questions or comments?
The question I have is for Mr. Skolnick. For me, in terms of the issue going forward, what I'm most interested in terms of the two different viewpoints here, is the predictability of one model or another. Do you have available any statistics in terms of the predictability in favor of using animals as models for disease or animals as subject for testing?
I don't have data to answer that question, but I do have this observation. There's little predictability, there's inadequate predictability. When they do a clinical trial of 5,000 people here, there's inadequate predictability of how the drug is going to work in the marketplace. Let's put it this way. I grew up with peanut butter and jelly sandwiches being served in school. Well, there's 3 million Americans who would possibly die from eating a peanut butter and jelly sandwich. The 6+ billion people on this planet are very diverse, and you have atypical reactions. Animal studies are not perfectly predictable, it's not that accurate as prediction, but that's not what animal models are mostly used for. They're used to give insights and generate hypotheses for clinical testing.
If that were the case, then a vast majority of grant applications to the National Institutes of Health would not end with these words. And we hope our proposed study will lead to a cure for X, Alzheimer's, stroke, Parkinson's, whatever. Okay? What this debate is in large part about is about what I consider to be the very disingenuous attitude on the part of animal modelers and their representatives who say, on the one hand, this isn't about predictability, because they don't have the data. Okay, I've got the data that shows that animal models are not predictive. No, what this is about is using animals as heuristics. And I'll grant you, if you want to get a new idea, you can do that in a variety of different ways, and one of those ways is to study animals. But if you're going to take the American taxpayers' money, and if you're going to take the March of Dimes money, and if you're going to take these other charities' money, then be honest and say, I'm looking for new ideas. And look, there's nothing intellectually inferior about looking for new ideas. I probably never had a new idea in my life, okay? But let's not be disingenuous and say, when we're looking for new ideas, we're really looking for the cure for cancer, because that's the only way they get the grant money. And again, what you're hearing here today are a lot of facts being thrown back and forth, and you're hearing a lot of just, completely opposite statements. And I would encourage you to read the books. I don't make any money from the books. All the proceeds from the books go to AFMA. But read the books. Look up the references if you have the time. But if you don't have the time to do any of that, just use some common sense. Think of the things that I said during my portion of the debate that are kind of theoretical and sciencey and somewhat ethereal. But that's the core of the argument.
Do we have another round on this question?
Yes.
I could give some examples of very important predictability. Let's take a look at 1960s. Some farmers, turkey farmers in England, had just devastating loss of turkeys. They were dying of what they didn't know what to call it, but they called it turkey X disease. Turns out what it was, after much research, using animals, using birds, it wasn't just turkeys, ducks, and other fowl got the disease. What it was, their liver was destroyed by a liver toxin. That was produced by a mole that was growing on the feed, aflatoxin. Well, sure enough, epidemiological studies later found that aflatoxin is a human carcinogen. It is causing a large number of liver cancers, primary liver cancers in humans. And it's something that now they're trying to keep out of our food supply. Now, another more recent, in the 90s, it's about 10 years ago, they found that cyanobacteria, blue-green algae, produce a toxin. And this toxin is liver toxic in animals and carcinogenic. And lo and behold, they did epidemiological studies in China, Africa, and other countries where the main water supply comes from surface water. And it is carcinogenic in human beings too.
Mr. Skolnick, if I can comment, I interpret those kind of examples as number 7 on one of the very first slides. I interpret that as animals as a modality for ideas, as opposed to what I was asking about in terms of animals as test subjects.
Please let Dr. Grigg comment first, and then you have a chance to follow up.
I think if you take nothing else from this debate, take this example. Yes, there are animals who get cancer from the same chemicals that give humans cancer. There's no question about that. But when a scientist says the word predictability or predicted, that has a very specific meaning. Now, the last time I checked, and these numbers undoubtedly have changed, but the last time I checked, there were over 2,000 substances, chemicals, that were found to induce cancer in rats and cats and dogs and mice. You know how many of those substances cause cancer in humans? Last time I checked, it was 30. Let's say it's doubled. That's not predicted. To say, I could go out and I could bet on next year's World Series. And I could place a $10 bet on every single team that's playing in the National and American leagues. And I'd be right. One of those teams would win. But I did not predict the winner. Okay, in science we have things like sensitivity and specificity and so on and so forth. And that's what prediction is all about. And I said, I think in my first talk, I said, you can almost always go back in time, retrospective, and find some animal that will react to a chemical or drug or disease or whatever, pretty much like the human does. That's retrospective analysis. That's not predictability.
One more round with us? Or you want one minute each?
What I said was that they didn't go back and test turkeys for aflatoxicity. They didn't go back and test animals to see if they also get cancer from pond water. These were the canaries that alerted scientists of the problem in human beings. They predicted. Now, I said in my talk that animal research is not primarily used to predict human reactions. They're used to provide insights and generate hypotheses that are tested in clinical trials. Now, we can't give people a suspected carcinogen. Dr. Greek kind of misrepresented. We know there's about 90 pretty strongly linked, pretty strongly implicated carcinogens for humans. Maybe 30 that have been definitely confirmed. But there's thousands that are suspected because of the animal studies. We can't do the studies to find out if they are indeed carcinogenic in humans. It's unethical. So there's nobody, except maybe Dr. Greek, thinks there's only 30. There's a hell of a lot more. The same thing with teratogens. Many suspected ones, only about 20, 30 that are known to be teratogenic in human beings, causing birth defects. We can't do those studies. We can only infer from the animals because we have no better means.
It's actually easy to do those studies. It's called epidemiology. And yes, there are only 30 confirmed. And again, maybe it's 40, maybe it's 50, 60, it's irrelevant. The point is that if the turkey is going to be predicted for carcinogenicity testing, what you have to do is that you then have to take those known human carcinogens, all 30 of them, give it to the turkey and see how the turkey fares. And the turkey doesn't do well. Okay? That's what drug testing is all about. You have an animal that, quote, according to Mr. Skolnick, predicted thalidomide's toxicity. Okay? Thalidomide was a drug that was toxic to humans when given to pregnant moms, and we did find the white New Zealand rabbit reacted the same way. Well, guess what? The white New Zealand rabbit was then exposed to a lot of other known human teratogens, and those known human teratogens passed with flying colors. That's the difference between predictability and retrospective analysis. The gentleman was right. You can get ideas from anywhere. And look, if I see cows dying in the field, I kind of want to know what they're eating. And then, yeah, let's see if other humans have eaten that.